Okay, today I'm going to talk to you about current BMI projects, which was not my choice of titles, but I'm going to attempt to fit my material into that title. So BMI, of course, is the Department of Biomedical Informatics. And I just want to give a couple quick updates, just to point out that we have 3 new faculty members. Dr. Skylar Resendez, who was formerly a postdoc with Dr. Elkin, who has experience in clinical informatics, some AI, some gene expression analysis, and did his PhD actually in evolutionary biology. We have 2 starting faculty members who work on AI, Dr. Zhao Zhang Lu and Dr. Sanjin Sang. They're actually a married couple. Dr. Liu theoretically started on January 31st, although I've been told that or something. She works on AI ML for omics analysis in chronic disease like Alzheimer's. And Dr. Sang works on AI and ML for image analysis, and he's starting, I think, June 1st. He's currently at the University of Miami, although they're both actually originally coming from Stanford, where they're doing postdocs. Dr. Werner Koisters had a wonderful postdoc, Michael Ravenberg, who unfortunately, I think, left to take recently a job in industry. I want to point out that we have 1 open postdoc slot under a T15 training grant, which is open to U.S. citizens and permanent residents. So if anyone's looking for a postdoc, we also may have 1 or 2 open T15 training grant slots for PhD students starting in July 2025. That depends on if people who are scheduled to finish in time. So I'll talk briefly about my current funding. I've been on the UB CTSA grant for many years, 5% effort. I've used my past support for development of the magnetic resonance imaging acquisition and analysis ontology, which is part of the imaging informatics goals of the CTSA grant. And my colleague, Alex Bartnick, is actually going to present a talk immediately following mine on that ontology in particular. I'm currently on a project called the Ontology of Gentle Fear and Anxiety, 12% effort. That's spearheaded by Dr. Bill Duncan and Daniel McNeil of the University of Florida School of Dentistry. And it's part of this larger group of behavioral ontologies and networks, which there's this transcendence ontology I've heard about. In the past year, I've been getting involved in another project called SOLCAP. And SOLCAP means Standardized Ontology Unique Labeling for Cytometry Annotation of Populations, which was a lot of work. I didn't create the acronym, but this is a group of investigators who work on flow cytometry, which is, of course, a technique to look at the cell surface expression of different proteins on different cells, as well as sometimes internal proteins. And it's used very much in immunology research, but also in clinical settings, in disease, for diagnosing disease. And so this is a complex project. It's was organized by Ryan Brinkman, and some of you know Ryan, perhaps. I think Barry might remember him. So Ryan is a flow cytometry informatician who has been involved in past in the development of OBI, the Ontology for Biomedical Investigations. But he was formerly a professor at the University of British Columbia. He now works for a company called Dotmatics. He's assembled a huge consortium of academics and industry partners. And there are 3 main working groups. The annotation working group is using information from various experts to create, using a huge spreadsheet, creating a representation of flow cytometry gating strategies and marker cell type associations. This is led by Kelly Lunston, but there are many experts involved. There's a bioinformatics group, and this is using an algorithm developed by Ryan Brinkman and Justin Mescas to, it's a way to normalize flow cytometry results from disparate sources from different institutions or using somewhat different sets of antibodies. And the idea is to come up with a signature to identify particular cell types. And so where ontology comes in here is that we're going to use the results of the annotation working group to enrich the cell ontology representation of immune cell types. So I've done a lot of work and others on these cell types in the past, but we recognize that there's new information. There are cell types we don't properly represent at this point. And there's also a problem with the cell ontology in that we're also getting a lot of results from single cell RNA sequencing, and we have to have a way of properly aligning those results with the flow cytometry results and associating them with the appropriate cell types. I also hopefully will work on something called the antibody ontology, which I worked on in the past until we ran out of money, and the antibody panel and gating ontology. And this is to represent the antibodies that the annotation group say are needed for their particular antibody panels to identify certain cell types and to describe the gating strategies. And again, the point of this is to standardize information here. I'm not going to get into too much detail about it, but it should be in advance. So as I said, Ryan has assembled a huge number of researchers and representatives of industry. Different companies have signed on to this project. So we hope that it goes somewhere. I'm part of a grant that was submitted in August to support our work on this. This is just one grant of several that are planned, but hopefully we'll hear about this in a positive way if the study section is ever allowed to happen, which is supposed to be in March. I don't know if it's still going to happen then. Okay, so, and that will provide some support for me and a grad student here at UB. Okay, so I'm going to just be going over a series of other projects here. So this is the ontology of dental fear and anxiety, which aims to represent entities associated with why people are fearful of dental treatment and their related behavior, which can include, of course, never bothering to go to the dentist or making an appointment and not showing up, or just developing fear or anxiety related behavior related to going to the dentist. This is going to aid in analyzing survey data, some existing survey data from several different sources, as well as new surveys that we're going to be doing that we're going to design in the second and third year of the grant. Again, this is Bill Duncan, Dan McNeil, who is a psychologist and has worked a lot in this field in the past, Asta Singhal, who's actually a dentist, and Olga Entz, who's also a dentist. And from the University of Utah, we have Brenda Heaton, who's a public health informatician. And I've been working on early aspects of the ontology development at UB with help from Ram Challa, who is trained actually as a dentist in India, but has come to do a master's in biomedical informatics and turns out to be quite talented in helping us in the early stages of understanding dental fear, since he's seen it in person. And then also Elena is now going to be working on us to start looking at some of the data dictionaries for the surveys to, again, try to figure out how to represent them ontologically. And so as part of this work, we're developing, so we have the ontology of dental fear and anxiety, but we're developing related classes that will actually go into the oral health and disease ontology, which is an ontology that Bill Duncan and Alan Ruttenberg and many others worked on starting over a decade ago, but is still an ongoing project. So another project here is the pain ontology. This is, again, it's a working group led by Bill Duncan. I've been involved, though, pretty much since the beginning. Finn, who's in the room, has been involved. I have a very talented undergraduate, Ava Cunningham, who's helping us on a specific set of terms, which I'll talk about on the next slide. We're working with a pain expert, Roger Filligim from the University of Florida. We have Matt Diller, and somebody who's recently joined the project is someone named Bronwyn Freeman from Germany. I don't fully understand her background, but she seems to know a lot about pain, which is a good thing. We also are developing a grant here, and this is not quite the same group because we're including Justin Reese from Berkeley National Laboratory and Emily Bartley, who's another pain expert who's from the University of Florida. And so a project I'm working on, or really my student Ava is working on, is this turning this international classification of oral facial pain, which is a terminology of entities related to oral facial pain, into a proper ontology. So Ava has basically taken the definitions, and there's, I mean, almost 200 terms in this terminology. It's really quite a nice terminology, and tried to, break them down, improve them, the definitions into more of a like Aristotelian format, but also identify additional elements that we'd want to represent, through linkages, logical axioms, and so on. So, for instance, we have something like pulpal pain due to hypersensitivity, which is a subtype of pulpal pain, which is so pain here is a process, and it's causally related to a disorder of the dental pulp. And of course, that disorder is located in the dental pulp since there's two material entities there. But we have the hypersensitivity. It's also a realization of this disposition, which is hypersensitivity to pain in the dental pulp, which is a subtype of hypersensitivity to pain, which is a disposition. And dental pain itself is a subtype of oral facial pain. So we're going to be building up a network here that relies on a variety of external ontologies as well as the terms that are in the ICOP itself. Okay, switching gears again. My group for several years has worked on something called the protein conformation ontology, which represents protein secondary, tertiary, and quaternary structures as conformational dispositions. This is based on the fact or the idea that many proteins or parts of proteins have the disposition to fold into a range of conformations, meaning that, so the traditional idea is that the protein primary structure, its protein amino acid sequence, is determinant of how it's going to fold up. But we know in practice that in many cases, under different environmental conditions, this folding will be altered. And in the extreme case of protein aggregates, such as prions, you can have completely alternate conformations. But even in the functioning of things like cell surface receptors, like the voltage-gated sodium channel or the epidermal growth factor, we can model the different conformations and the conformational changes that the protein or the proteins involved go through in terms of during the activation of that receptor. So this has been worked on by originally an undergraduate, Morgan Mitchell, who's now a UB medical student. And then Giacomo stepped in and provided a very nice representation of the conformational changes for the epidermal growth factor, which was interesting because they had to rectify two different models, which both may be true, but may be occurring under different environmental conditions, for how that receptor binds its ligand and then dimerizes and then transmits a signal. Also, Lauren Wishney and Bill Duncan have contributed to some of the work here. We submitted a paper on this in late November, although I will also mention that Giacomo presented a shorter paper at a meeting whose name I can't possibly remember because it was very complex, but it was in Greece, which is why it was a good meeting, I suppose. And that was last May. Okay, so there's also the neuroelectronic interface ontology, which is also known as the bionic vision ontology, which is a crazy name. It's an ontology that represents initially different bionic vision devices in the eye diseases and disorders that they are used to treat. So we have two experts on our working group, Daniel Rathburn from the Henry Ford Institute in Detroit and Michael Byerly from UC Santa Barbara. Again, working with Bill and Sam Smith, who some of you may remember, who's worked with Oliver He in the past. He's kind of retired, but still sort of active. From UB, we have Jisoo, and some of you may remember Patrick Ray, who was a UB philosophy student, finished probably 7 or 8 years ago. Patrick had worked with me for a period and created an ocular disease ontology, and also wrote a very nice paper describing blindness in a sort of ontological fashion. And both of these works kind of have had some influence on how we're developing this work. We're currently writing a grant, which kind of in early stages, we're kind of deciding exactly how we want to focus the grant. But so I've been acting as more or less an unpaid consultant to the Kidney Precision Medicine Project. So there's a couple different parts of this. So there's the larger group, which is illustrated by this paper, whose title is at the bottom, and you can see the large number of people there. And this paper kind of describes their overall ways that KPMP is using ontologies to represent and analyze data. And Eddie Muniz, who's sitting over here, he's a UB master's student in biostatistics and bioinformatics, has been working with me to do analysis of data generated by KPMP, single cell sequencing data from patients with acute kidney injury and chronic kidney disease, as well as normal controls. And so this data is already associated with terms from the cell ontology. And we're using gene ontology term enrichment to identify changes in biological processes between AKI and CKD and normal controls. So there's a few interesting things here. Acute kidney injury, which is sort of a, obviously sounds acute, so it happens very rapidly. In this, we see a downregulation of a lot of normal, like transport and metabolic processes that happen in the kidney. Whereas in chronic kidney disease, actually we see upregulation and also upregulation of some kidney development terms, suggesting there's probably some attempt at regeneration going on there. So we do have this nice figure, which may be money making. So the idea here is that this is just looking at the expression of a single gene and a number of cell types. The cell types are in the rectangular nodes, and I know you can't see it, but it goes normal, AKI, and CKD, and the darker the color, the higher the expression. So we can see that the expression of this particular gene or protein SPP1, which is actually a known marker for kidney disease, is particularly upregulated in acute kidney injury and not nearly as upregulated in chronic kidney disease. And these cell types here are tied to the anatomical locations where the cell types are found. And those anatomical locations, of course, we can map back to the actual structure of the kidney. Okay, so another project here is the cholangiocarcinoma ontology, which was the work of my PhD student, Anawat Pengput, who finished in May 2024. The ontology represents different subtypes of this cancer, cholangiocarcinoma, but more importantly, it has classes that allow us to analyze demographic and ultrasound screening data, as well as EHR data, which I forgot to write down. So cholangiocarcinoma is a cancer of the bile duct that is caused by a liver fluke that infects people who eat raw fish that is already infected with this liver fluke. This is a disease that's found very much in Southeast Asia and Thailand, and there's not surprisingly been many public health campaigns and studies in Thailand around this disease. Anawat came to UB to do a master's. He worked previously as a public health officer in Thailand and was involved in some studies related to this disease. And so that was part of the reason he chose this project. So the goal here was to identify risk factors for the development of cholangiocarcinoma. So we used the term enrichment technique, which similar to what we do in the gene ontology here. And this is a very large ontology, which is really covering a couple different domains, but we have verbal screening results, ultrasound screening results, and the diagnoses from the electronic health record systems. And so what we're doing here is comparing the 186 patients that were diagnosed with cholangiocarcinoma who took part in earlier studies where they did verbal screening for different demographics, lifestyle factors, education level, and ultrasound screening, which was done on a large population scale. That was done 1 to 5 years previous to the patients showing up at the ER. So we had to match the patients through their Thai social security numbers, which we had to do in we had to de-identify the data, basically was the first step, which NCOR did. I never saw those numbers. And once we did that, we hid the original data away for the IRB. Okay, so we can consider here that these 186 patients compared to the population as a whole, they're overrepresented for reporting alcohol consumption and smoking. And again, that's not a complete surprise since, again, people who smoke or drink are often more resistant to public health campaigns, education campaigns, and so they may have resisted campaigns to tell them not to eat raw fish. Overrepresented classes for occupation include farmer, people being of the male gender, and people having diabetes and a few other factors here. And then when we look at the pre-screening, the ultrasound screening that was done again 1 to 5 years before, there were certain characteristic features that these patients who ended up having cholangiocarcinoma had, such as periductal fibrosis, of which there are 3 types, and the type 3 is the most diagnostic. But things like mass in the wall of gallbladder, cirrhosis, liver parenchyma. So these patients were clearly, this particular set of patients were clearly already a bit sick, could be measured on ultrasound, though they were not diagnosed with cancer at the time of the ultrasound screening. But they were picked out, many of them were picked out at the time of ultrasound screening as having suspected cholangiocarcinoma. They also showed signs of weight loss and jaundice, and there's some other features. But what NCOR did was then take these overrepresented classes and use them to drive a machine learning analysis to identify the important features. And then I asked him to remove all the top features and see how well some of the minor features could perform in this model. And so, I mean, again, this top features included things like dilated bile duct or periductal fibrosis or mass in the liver. But when we move some of these top features, then other things come to fore, like the occupation and education level and alcohol and smoking. And so this suggests that if you didn't have enough money to be screening people with ultrasound, you could do demographic surveys and get a lot done here. So other projects that I remain involved with include cell ontology. I continue to participate in development. And I'm planning a sabbatical in the fall of 2025 with David Osumi-Sutherland, who's at the Sanger Institute outside Cambridge, which is across the street from EBI, which was mentioned earlier. And I'm going to work on immune cell types again, which I've done before. I still work with the gene ontology kind of as a consultant for immune system related processes, which basically I look for GitHub issues where they're trying to change the term that I created. And I say yes or no. I often get annoyed, but that's okay. I teach about the Go and biochemistry 419, 519, which is co-listed now as a BMI course. I've done this for 12 years. It's the only teaching about ontology that I get to do, so it's fun. There's the immunology ontology, which is a long-term project. We've had periodic meetings and we've made some early steps. And I've spoken quite a lot over the years with a guy named Aaron Abend, who runs this thing called the Autoimmune Registry. And we actually spoke to him a couple of months ago. We're going to work on a grant to fund a workshop for autoimmune common data elements related to ontology. So now, the last ontology I'm going to mention here before Alex gives his talk on it is the MRIO ontology. The MRIO is probably a good acronym because it's about MRI. And if you know what MRI is, so we published a paper on this in neuroinformatics last April or May. I guess it came out in May, really, but and Alex will give a lot of details on this. I'll just have this as my last slide. So we met a guy named Carl Helmer at ICBO in 2022 who was developing a similar ontology called NIDM, which is like neuroinformatics something, data management. Yeah. Right. So we had this idea we'd like to work with him, in this beautiful ontology world where we bring people together. And so we're going to create a new ontology if we get the grant that we just wrote, which will be called the Image Ontology for Multimodal Biomedical Applications. So it's going to have a broader scope than MRIO. It's going to cover other types of biomedical imaging. And part of the reason we did this is that we developed some upper level classes that have been contributed to the Ontology for Biomedical Investigations, which are related to imaging. And these are some fundamental classes. And so we think this is going to enable others to work with us as well as maybe we ourselves to add different modalities like CT scans and confocal microscopy. And so terms to represent the methodologies involved in those things. And we work quite closely with the OB developers. And a lot of our classes from MRIO have actually been transferred to OB because, in particular, Bjorn Peters was a great advocate that these should be more widely available in OB. And getting them into OB means that they're going to persist even if we don't. So, so with that, I'm going to stop and let Alex take over. Although I suppose maybe I should take a question.